So after the fertilization of the oocyte, we talked about the first phase of embryogenesis, which is the phase of the formation of the cellular blastoderm. Then now we'll talk about the second phase of embryogenesis, which is the gastrulation. So in different organisms, gastrulation is important for the formation of the three embryonic layers: ectoderm, endoderm, and in between the mesoderm. So here in Drosophila, we'll talk about each of those layers and how they are formed during gastrulation. Regarding the mesoderm, okay, it's called prospective mesoderm because it will give in the future, these cells will give in the future the mesoderm. So the prospective mesoderm is about 1,000 cells. So this is the Drosophila embryo at the beginning of the gastrulation. What we see here is the ventral side. From the opposite side, we have the dorsal side. So here we are looking at the ventral side, and at this stage, so we see at the ventral side a kind of furrow called a ventral furrow. Okay, so this ventral furrow is formed after the accumulation of 1,000 cells here in this midline called the ventral midline, and these 1,000 cells accumulated at the ventral midline will be invaginated, or they will be folding inward, it means to the inside, in order to form a kind of ventral furrow. This is the ventral furrow. It's a kind of canal formed at the midline ventrally, and this ventral furrow will be pinched off. It means it will be detached from the surface to the inside to become a ventral tube, and this ventral tube will be differentiated later into different mesodermal organs. So we are still looking at the ventral side, but now we'll talk about the endoderm, the second embryonic layer. So after the mesoderm, we'll talk about the prospective endoderm. So here we'll see during the formation of endoderm, we have small invaginations like pockets. Since they are small, very small invaginations, we have one small pocket right here. It's the anterior pocket at the anterior end, and the second pocket is formed at the opposite end, a bit dorsally. So if we turn this embryo from the ventral side, on the other side here we see the dorsal side. So ventrally we can see the anterior invagination, the anterior pocket, and a bit dorsally we can see another pocket. Okay, it's the posterior, at the posterior end. Okay, so through this pocket cells will invaginate. Okay, and in addition we have the pole cells. They are localized here at this stage. We mentioned last time that the pole cells will give the gametes. Okay, pole cells will be internalized along with endoderm through this posterior pocket. So it means they will enter to the inside through this pocket. Okay, they won't stay outside the body. Okay, by this stage they will enter to the inside, and at this time the embryo has a bending form. It's like this. It's not a straight embryo. It's like this. You see it? It's like that. Okay, we have a dorsal bending of this embryo to form here a kind of furrow called the cephalic furrow. Okay, called the cephalic furrow, and this cephalic furrow will just separate the future head from the rest of the Drosophila body plan. So we talked about the prospective mesoderm, the prospective endoderm. Now we'll talk about ectoderm. So ectodermal cells are at the surface. Okay, so these ectodermal cells on the surface, they will undergo a movement, and this movement is called extension. Okay, it's a kind of extension movement in order to form a germ band. Germ band is this purple structure, okay, formed by ectodermal cells, okay, and this germ band will contain the cells that will form the trunk of the body, okay, or the trunk of the embryo, okay. This is the head, it's called procephalon. This is the head and this is the trunk. So the first phase in ectodermal formation is called germ band extension. So this cell movement will bring the most posterior segments of the body, okay, just behind the head. It means just right here. So this is the future posterior side is located here, okay. But after, of course, these cells should be relocated at the posterior side. But during this phase, we have a germ band extension. So this will bring the cells just behind the head. Okay, so these posterior structures will be located at this period behind the future head region. So first we have a germ band extension, and this germ band extension, okay, you see it here. During the germ band extension, we have different morphogenetic process. It means we have different modifications in the Drosophila embryo that will occur, like we have organogenesis. We have the formation of different organs in miniature, okay. Like for example, we have the segregation of the imaginal discs. You know, in Drosophila we already talked about imaginal discs. They are the structures that will give the adult organs, okay. They are called in the larvae and in the embryo imaginal discs, okay. We also have, during this germ band extension, we have a segmentation. You see, it's clear that we have the head segments, 3 head segments. We have 3 thoracic segments, 1, 2, 3. We have 8 abdominal segments. We see 1, 2, and the other ones are dorsal, okay. This is the ventral side, this is the dorsal side. Are dorsal, this is the abdominal 1, abdominal 2, 3, 4 to 8. The abdominal 8, through the germ band extension, is brought just behind the future head, okay. And this is the germ band right here. So this germ band extension is followed up by a germ band retraction. You see, we had an extension followed by a retraction. So you see the A8 and the other abdominal segments are now at the most posterior part of the Drosophila embryo, okay, are at the posterior side through this germ band retraction. So germ band extension, germ band retraction, and the third movement is the migration of those ectodermal cells dorsally to cover what do we call the amnioserosa, okay. They are embryonic layers, okay, like amnion. You know, even a human embryo have amnion around it's an extra embryonic layer, okay. Amnion and the serosa, they are fused together to form amnioserosa. This is the amnioserosa. It's located dorsally and it should not be naked. It should be covered by ectodermal cells. That's why we have movement of those ectodermal cells dorsally to cover up this amnioserosa, okay. And this is due to the interaction between those epidermal cells and this amnioserosa to encourage, to help out the migration of these ectodermal cells dorsally. And once these epidermal cells have been migrated to the most dorsal side, we talk about a dorsal closure, okay, a dorsal closure. This process happens during the gastrulation. By the end of the gastrulation, it's called dorsal closure because it will close the dorsal outside of the embryo by the fact that ectodermal cells will cover completely the amnioserosa. So I will show you the three movements of ectodermal formation by the following video. So you will see how the germ band extension will happen, how the germ band retraction will happen, and how this dorsal closure process happen in a live embryo. Okay, I will show you this in a video. So by the end of gastrulation, we have mesoderm, we have endoderm, we have ectoderm. You see, this is the mesoderm. It's ventral. It will give the muscles and the connective tissues. Then we have the endoderm. The endoderm is usually will give the gut, okay, in yellow. And here, regarding Drosophila, the gut is divided into three parts. We have the foregut at the anterior side, we have the hindgut at the posterior side, and we have in the middle the midgut. Here you should know that the endoderm will only give the middle part of the Drosophila gut, called midgut. Okay, the midgut, while the ectoderm will give the foregut, the anterior gut, and the hindgut, the posterior gut, plus of course the ectoderm in different organisms will give nervous system and epidermis. Here you see the nervous system in insects. It's the opposite of what happens in vertebrates. In vertebrates, the nervous system is dorsal, while you see here in blue, the nervous system is ventral. Okay, so nervous system, it's an ectodermal derivative, foregut and hindgut as well, and also the epidermis. Okay, what you see here in white. Okay, the epidermis is usually covering the embryo from the outside. It's an ectodermal derivative.